now ibrutinib is like a multi-billion-dollar drug. And, like to think that the dog had like a little bit to do with at least getting them there a little faster, which is kind of cool.
Hello, and welcome to the Veterinary Cancer Pioneers Podcast, the show where we delve into groundbreaking work of veterinary professionals dedicated to advancing the field of veterinary oncology. I'm your host, Dr. Rachel Venable, and I'm thrilled to embark on this journey with you. Today's episode is brought to you by ImpriMed, pioneers in AI-driven precision medicine for veterinary oncology. Their personalized prediction profile helps you make confident treatment decisions for your canine lymphoma and leukemia patients by predicting how your patient could respond to multiple chemotherapy protocol options. Learn more at imprimedicine.com. That is IMPRI medicine.com. Hello and welcome everyone to the podcast. I'm so excited today. We have Dr. Doug Thamm, and honestly, to go over his full bio would take half the podcast, so I'm just going to hit all the highlights today. So Dr. Thamm, he is the Stuart Presidential Chair in Oncology and Director of Clinical Research at Colorado State University Flint Animal Cancer Center. He's authored over 200 publications and 25 book chapters, and he's also the co-editor of Withrow and McEwen Small Animal Clinical Oncology, which is basically the grail for veterinary oncology. And he's also the editor-in-chief of the Journal of Veterinary and Comparative Oncology, which is also a huge journal in veterinary oncology. And he's the president of the Veterinary Cancer Society. So definitely has his pulse on a good leadership role, knows a lot about veterinary oncology right now. And he was actually the 2023 recipient of the Alan Kelly International Prize in Canine Health from the Kennel Club. So you've done so much, Dr. Thamm. I'm so excited to kind of dive in, learn more about your journey and just what you guys are doing now and where you think the future is heading. So thank you so much for being with us today.
Oh, my pleasure. Thanks for having me.
Yeah, so I always like to start just to get a little bit of people's background, kind of what got you into veterinary medicine or certainly what got you into veterinary oncology.
I think we all can appreciate with these kinds of people, but I grew up being one of those animal kids. So we had dogs and cats in the house, and I was always bringing home little creepy crawlies that I found outside. And I think in my sort of early teenage years, I think I maxed out at maybe having 7 fish tanks plus a pond in the backyard. And I was also like a little equestrian kid too. So I, owned a horse in high school and did all that. So, had my feet in the large animal thing, the small animal thing, and the exotic thing, like basically all through high school. So, honestly, I couldn't really think of another thing that I wanted to do as a career. Although I had a little bit of a crisis in high school and college where I thought I was going to sort of do music for a living instead of being a veterinarian. But then some people talked some sense into me and said, hey, you can always be a veterinarian and do music on the side. Can't be a musician and do veterinary medicine on the side. So I kind of made the decision for me. But yeah, it was just one of those sort of like logical things, just because most of what I did in my spare time had something to do with animals one way or the other. So why oncology is a little bit more interesting. So even as an undergrad, like I kind of had the research bug already. So I worked in a research microbiology lab through a lot of undergrad. And, if I didn't get into vet school on my first try, I was going to go, do graduate work in immunology. So kind of always thought that the researchy component of this career would be really, really cool, and kind of knew that I probably wasn't going to be happy, sort of being in sort of primary care and sort of doing that. Don't get me wrong, that's an amazing way to make a living, and the relationships that you build with your clients are so wonderful and all that. And we desperately need those people. But, I was really worried that I was going to be like bored out of my skull after 45 minutes in sort of primary care practice, but didn't really know for certain sort of what the specialty I wanted to do was. So over a period of time, thought I wanted to be like a large animal surgeon, and I wanted to be an exotics doc, and wanted to be all kinds of things. And one of the things that kind of really sort of focused oncology for me was a personal cancer experience I had. So after my freshman year of vet school, I was actually diagnosed with non-Hodgkin's lymphoma. Had to take a year off, get treated for that. And I never want to give people the impression that it was like, oh, it was a life-changing experience. And that's when I decided I was going to dedicate my life to curing cancer. Like, it definitely was not that kind of experience. So I went in with a 97% likelihood of cure based on the way I presented. So it was really like an enormous inconvenience that I had to take this time off and get treated for lymphoma. But I hadn't really even given oncology a thought before that. And just that personal experience made me go, wait, huh. I mean, I'm only through 1 year of vet school, but I haven't learned anything about oncology yet. Like, I wonder if that's a thing. And just kind of started looking into it, just kind of as a result of this unfortunate event that happened. And I was like, hey, this is really cool. And like, it does seem like there's lots and lots of research opportunities, sort of specifically in oncology, and, maybe even more than in some of the other disciplines. So maybe I could scratch my research itch pretty easily if I ended up doing oncology. And I don't know, the rest is history. Although there was a very brief period of time in my 4th year where I revisited the fish thing and almost pivoted into fish medicine. But the kind of internship I was trying to finagle didn't end up working out. And here I am.
I feel like for the veterinary oncology community, I'm glad you didn't go into fish.
I know that.
glad you-
What a loss for the fish community, right?
I guess so. I don't,
I could be
but,
the editor of the Journal of Fish Medicine.
I'm sure there's a handful. Some
Yeah,
people that
there
read-
totally are.
No, that's great. But and I'm so sorry that you had to go through that Hodgkin's lymphoma, but I'm glad that it all seemed like it went as well as it could, for
Yeah, I mean, don't get me wrong. It was a drag. It was a hard 10 months, but I mean, it's one of the things that got me where I am. So that's all right.
Have you found with your experience, like how we treat animals and how they treat people, do you find that they're just even in the clinical setting, is there a lot of overlap or is it very different?
It's really neat, actually. So one of the things that I had an opportunity to do as part of my clinical training, during my residency, was actually do an out rotation in human oncology. And I actually went back to University of Pennsylvania, where I was treated, to actually do that out rotation and actually sort of, rotated on, a bunch of the clinics for a couple of weeks. I was really kind of struck at how similar the conversations were that were being had with clients, with patients there, compared to the conversations that we have with our clients, with one very, very specific and huge difference. And that is, there was no talk about money that happens, in the human oncology clinic. It's all about what's treatment going to look like and what are the side effects going to be and how well does it work and all those kinds of things. And, we always sort of talk about, oh, we can never predict what's going to happen with an individual patient, and all the statistics that we have are in aggregate, and they have all those exact same discussions. But I mean, the thing that is so different is the amount of time that we spend talking about money. And the other thing that I think is actually neat in the way that I think we do a much deeper dive into sort of explaining the whys into what we do. Why are we doing this test? Why are we doing that test? Why are we doing this treatment? Because of the financial implications. Like, I feel like, for better or for worse, we have to sort of justify what we're doing in a way that they don't have to in human medicine, which also allows us to, I think, impart more information to our owners, whether they want it or not, about the whys, because we're trying to help an owner decide how to spend their money in a way that human patients, whether it's cancer or something else, usually, thankfully, don't have to do. So I think that's the biggest difference I noticed.
Oh, yeah, that is really interesting. I never thought about that aspect, but that makes sense. When I've gone to my doctor, they've never talked to me about prices, so.
Yeah. And they probably haven't talked too much about the whys. I mean, I think they might talk about the whys to us a little bit more because we're like colleagues, right? Or we might be more inclined to ask just out of curiosity. Hey, how's that test work? And, what are you looking for? And those kinds of things that like a regular patient probably wouldn't. But yeah, I mean, in your day-to-day stuff, that stuff just doesn't come up.
Do you think, since money doesn't really come up, do you think that we give more options as well? Like, I feel like, generally speaking, when I'm talking with, new pet owners, I usually give almost 2 or 3 options. Like, I always try to give more than one,
Yeah, I mean, that whole concept of spectrum of care is something that I don't think comes into play as often. I think on the human side, I think the exception is probably in geriatric care, right? Where you've got all these comorbidities and all these other things that are going on. And there, I think maybe there's a little bit more of the, well, we could do this, or an alternative could be this less aggressive thing, given the fact that, you're in cardiac failure and renal failure and you've got that diabetes and, like all of those things. I mean, that's one of the things that I talk to my residents all about is sometimes we get these really, really complicated cases, that not only do they have lymphoma, but, they have all these other medical things going on. They have unregulated Cushing's disease and they've got horrible skin and they've got whatever. And I'm just like, dude, this is what every oncology patient is like in human medicine, because they all have like 3 or 4 other comorbidities and 3 or 4 other specialists that they're seeing. So it's just like, be glad that we don't see that more often than we do. But yeah, that's another thing that I think is so different is there's usually so many other things wrong with human oncology patients that they're trying to manage that I think less frequently we're having to deal with.
Yeah, I hadn't thought about that either, but you're right. Because a lot of times it is in older people and yeah, you're right. They probably do have so many comorbidities. So I am glad that a lot of our patients don't have that. And
Yeah.
speaking about people, you've done a lot of research looking at comparative oncology and things. What's some of the most interesting or the most exciting that you've done that's really been a good comparative example with people and dogs?
I mean, I guess there's one sort of old one that comes to mind, and then there's kind of a newer one where the jury's still out on how comparative it's going to be. But one of the coolest ones that we were able to do was the early work that we did with ibrutinib, which is, a small molecule inhibitor of a kinase called BTK. People are starting to use it in veterinary oncology these days for the treatment of B-cell lymphoma. And I don't know, it's probably like 12 or 14 years ago, we actually started looking at it as a therapy for dogs with lymphoma in conjunction with this little startup company from the Bay Area called Pharmacyclics, which was trying to develop it as a human drug. And at that time, like the main question that we were trying to answer didn't really have much to do with did it work or not. We were trying to help them actually work out an assay to know if the drug was actually sort of sticking to its target or not in cells. And the question that they wanted to ask is, can we look in the blood to see reliably what's going on in the tumor tissue? Because people don't mind having their blood drawn. They don't like having biopsies taken of their lymph nodes when they've got lymphoma, right? So we did both. We looked in the lymph nodes, we looked in the blood and said, hey, are the results that we got from this assay looking to see if the drug was sticking the same in the blood and in the lymph node? And yep, they were. So we kind of said, hey, that's great. So you can probably just look in the blood of your human cancer patients and feel pretty comfortable that same thing's going on in their lymphoma tissues. But the added benefit was like 3 out of 8 of the dogs that we treated in the first part of that study actually had partial responses to the drug, which was actually very, very unexpected. Because on the human side, this is mostly a drug that only works really well for the indolent lymphomas by itself. And, we don't see that many indolent B-cell lymphomas in our patients, right? So these were like your typical high-grade multicentric lymphomas. And the fact in retrospect that we really saw any activity was pretty exciting. And I'm not going to say that, our results was like the make or break thing that like rescued this drug or like stopped it from like ending up in the trash can or anything else. But I do think that the fact that this little company could go to their investigators and actually show data in dogs that actually worked in patients with actual B-cell neoplasia kind of, I think, got it into the clinic a little bit faster. So, a lot of these human lymphoma clinicians, they might have a choice between participating in 3 or 4 different clinical trials, and they got to decide which one they want to dedicate their resources to. I think the fact that Pharmacyclics, came to the table with data in dogs with spontaneous B-cell lymphoma probably got some people a little bit more excited about it than they would have otherwise been. So I think that was pretty cool. And, now ibrutinib is like a multi-billion-dollar drug. And, like to think that the dog had like a little bit to do with at least getting them there a little faster, which is kind of cool. So that's one of the older ones I think was neat. The newer one that's pretty exciting is this work that Steve Dow and Dan Regan have been doing, trying to repurpose the blood pressure drug losartan as a cancer therapeutic, specifically because of its ability to interfere with monocyte migration into tumor tissues. And I think you've had Steve on the podcast, so we probably talked about that. And it's really cool that I think we've kind of found this new way to use an old drug that might end up being effective in a completely different way than intended. And, I think we're really excited that this observation is now sort of trickling also into the human side. So there's a study in pediatric osteo that's going on right now looking at a combination of high-dose losartan with Sutent, which is kind of like the human version of Palladia, specifically in patients with metastatic osteo. And time will tell sort of how well the observations that were made here kind of translate there. But that's like kind of a newer example that I think we're kind of fired up to see how that goes.
Yeah, I think that is really exciting, the repurposing of old drugs, because I'm kind of thrifty by nature. So I sort of love the idea of getting to reuse things. But I also feel like for our clients, it makes something that otherwise might not be feasible, right? Some of these new drugs, I mean, they're just crazy, but something like losartan's very affordable, you know?
Yeah. And on the human side, it's the same thing. So there are different kinds of savings, right? If it costs $10 million to develop a repurposed drug versus $800 million to develop some brand new drug, those cost savings are going to trickle down to the healthcare system, if not to the individual patient, So yeah, I mean, it's important no matter how you slice it.
Right. And as you mentioned, I mean, the numbers can be astronomical. I think a lot of times people don't understand how much it costs to get a drug through all the process, from discovery to the actual shelf, where you can prescribe it. And you certainly know quite a bit about that. You've been involved a lot with Tanovia. Can you kind of walk us through what that process is like? Because I think most people really have no idea how much is involved.
Tanovia is kind of a neat example because, again, that also started with a human pharma collaboration. So once again, kind of like 12, 14 years ago, maybe a little bit more, we were actually hit up by Gilead Sciences. That's what they're called, Gilead Sciences, because they had this molecule that they were actually interested in looking at in people. And because of some oddities about Tanovia specifically, it turns out it falls apart in rodent plasma. So they couldn't use the typical sort of mouse models of cancer or of lymphoma that they would normally use to sort of test how well this drug worked. And they're kind of scratching their heads going, well, how are we going to figure out if this works? We can't just move forward into people without any preclinical data about efficacy. And the guy who was sort of running the program was a veterinary pathologist named Dan Toomas, whose daughter, funny fact, is actually one of our oncology technicians now. Bailey, but that's completely random. But Dan was like, well, like, I don't know, when I was a pathology resident back at Georgia, like we saw dog lymphoma all the time. I wonder if we could do, try this drug in some dogs with lymphoma. So he ended up reaching out to me and David Bale when he was still here. And we started these collaborations looking at Tanovia, but at the time it was to turn it into a people drug. And things happened that we're not privy to on the human side that made them decide, we probably don't want to keep developing this. And so for a period of time, it sort of sat on a shelf and nobody was going to do anything with it. But around that time, there was a guy here who started working for CSU Ventures, which was like kind of the discovery and commercialization thing that we have at CSU. This guy named Terry Opkenorth, who used to be an executive at Abbott Labs, which is now AbbVie, like one of the big old pharmaceutical companies. So he came in to sort of consult / run the show doing this sort of commercialization thing, and ended up being so interested in this whole dog oncology thing that he started this little company called VetDC. And he actually had some friends / colleagues at Gilead Sciences who he knew. And through some kind of backdoor channels, he was able to say, hey, what do you guys at Gilead think about giving us a license to the veterinary rights since you're not really doing anything with it anyway? And they said, sure, that sounds fine. They never would have said that to me, but, because this guy, kind of knew the right people to ask, he was actually able to get it done. And as a result, we were able to get not only the rights to turn into a dog drug, but just an astronomical amount of work that had already been done on the molecule in preparation for turning it into a human drug. So all the chemistry and the manufacturing and just reams and reams and reams of safety data and all that stuff that we were able to sort of repurpose into the application that eventually went into the FDA to get a license for the drug. So actually, all of the data that went into the application for conditional approval was data that Gilead gave us. So all the safety data, the efficacy data that was on the conditional label was all the stuff that we generated back when we were working with Gilead. The only thing that we had to redo from scratch was the chemistry, manufacturing, and controls. So how are you going to make it? Who's going to make it? How do you know that you're going to be able to make it the same every time? All that kind of stuff. So that was a huge pain in the butt, but at least we had a cookbook to work from because somebody else had already done it. So we basically had to redo it, but we were using somebody else's cookbook instead of having to start from scratch. So, I mean, that ended up being probably a grand total of like $8 to $10 million in savings. All that data that we got from Gilead. Then all that was really left officially in order to sort of get the drug approved was to do the FDA registration trial. So, the randomized placebo-controlled prospective trial that we did, that was published in 2021 or 2022 or whatever, that led to the full approval of the drug. But I think the smartest move that DC made was along the way doing all these other studies. So it wasn't just this one-off registration trial, but there were all these other studies that DC did, some of which they were able to sort of put some money towards, some of them which were really done just for the cost of the drug. But, at the time of full approval, there was 8 publications in the peer-reviewed literature about Tanovea and, how to use it and different groups of patients you could use it for and how well it worked and what are the things to watch for. So, I mean, I think the fact that DC came to the table with this giant portfolio of peer-reviewed published studies really, really helped for people to kind of understand it and sort of feel a little bit more comfortable with it early on, versus coming to the table where, I don't know, we haven't published anything yet. And all we can tell you is what's on the label. And that hasn't even been subjected to peer review yet. And that's what you know. So I'm really happy that DC seemed to be so excited about sort of generating knowledge, getting it out there for people to read, even in advance of, having a product on the market. So yeah, we were really very fortunate in a lot of ways, just in terms of who was helping us out and all the information that we had already had, sort of moving into it. And again, the fact that everybody was just so excited about doing the science and actually sort of generating the knowledge to help everybody sort of know how to use the drug. So that was really great.
Yeah. And how long would you say that whole process took?
Oh, I mean, from the first time we put it into a dog to the time it was fully approved was 12 years.
Yeah, it's long, right? I feel like a lot of people don't realize how much data, how much just time. And do you know roughly what it costs, or what would you say it generally costs to get something like that approved?
Oh, I mean, I think if we had to start from scratch, so if we didn't have all this information that was coming from somebody else that, really sort of foreshortened the process, I think you're easily into it on the veterinary side for conservatively, $8 to $10 million, which is a tiny fraction of what it would cost to do the same thing on the human side. But I think, when you think about that and the fact that, you're probably only making a few $1 million in revenue every year from these drugs on the veterinary side, especially these drugs that are, really only used by the specialists, I think all of a sudden it makes a little bit more sense why people are charging what they need to charge for them, right? Because really, they are just not making that much. And it costs a lot, by veterinary standards to actually get the drug to market. And, that's why I think more and more these days, just for purely financial reasons, I think a lot of the drug companies are saying, look, we cannot afford to develop a drug that's only going to be used by oncology specialists. We just can't. It's just a financial thing. They cannot make their money back on a drug that's used by 500 people, right? And that's why more and more, I think we're seeing the companies looking for either, immunotherapies, biotherapies, orals, things that, again, can be used at least partially by non-specialists and don't require special handling in order to use safely. And maybe that could be useful for multiple kinds of cancer. So I think those are the things that are really sort of guiding the decisions that a lot of these big pharma companies are making these days is, is this going to be used beyond just, the people with DACVIM after their name or not? And that's really what they need.
You make a good point, what, the ROI, right? What kind of return are they getting on these things? Have you found that some of these comparative studies where, we're studying a drug that's supposed to go into human medicine, but we're studying on animals with a natural cancer, and then if it goes into human medicine, it comes back to dogs? Does that ever really happen or not so much? Because is it separate licensure that they have to do?
Yeah, I mean, I know there's some examples where they're hoping to do that. So Massivet, which is approved in the EU for the treatment of mast cell tumors, they're still trying to pursue human clinical development of that. Stelfonta, they're working on pursuing human clinical development for that. So I think there's some people who are sort of trying to see if they're going to be able to be successful on that. But I'm not aware of anybody who's sort of crossed the finish line there where there's the human drug and then the exact same drug is being marketed for animals. And one of the things that I think makes people gun shy about doing that is this perception of differential pricing, right? So, and this is coming from the human side, right? So why should I pay $80,000 a month for this drug that you're selling, on the veterinary market for $800 a month? There's one really old example where this actually happened that caused a lot of problem. And that was like back in the 70s or 80s. It actually was discovered that levamisole was a potent potentiator of 5-FU-based chemotherapy in humans with colon cancer. So if you co-administered 5-FU and levamisole, it actually made the 5-FU work better. So they were actually developing, and they may even got to the point where they were actually selling levamisole as a chemotherapy drug for humans with colon cancer. Well, at the time, you could also buy levamisole in a 55-gallon drum at the feed store because it was a cattle dewormer. And the human patient advocates went crazy, and they're like, wait a second. Like, you're charging us, thousands of dollars for this drug that I can get at the feed store? And right, like legitimately so. So I think that's made a lot of people a lot more gun shy about this idea about the perception that's associated with this sort of differential pricing and differential marketing of the same drug, for veterinary and human indications. So I have yet to see that done successfully, but time will tell.
Makes even Palladia, because the other drug was sunitinib. I think I'm saying that correctly. So would you say they were still different enough that it's not quite the same scenario?
Oh yeah, they are different chemical entities. So they only differ by a single bond, but anybody who looked at them could say, yep, they're different. They definitely pass the smell test in that way.
Right. And the other thing you bring up about the cost differential, I've had people bring up before, actually physicians, where they'll say like, well, the chemo we give to people is different than what you give to dogs. But as far as I'm aware, it's the same drugs, right? Like they're not like a different grade. Like the doxorubicin we give is the same that they give to people, or is that not correct?
No, it absolutely is. So when you're talking about these older generics, yeah, they're totally the same, and the cost structure is exactly the same. So it's when stuff is still on patent and the drug companies can charge whatever they want, that it's really the biggest issue. And that's why so often in veterinary medicine, we're really limited to the generics, right? Or, now, of course, there's that whole sort of compounding route, which is very much, kind of caveat emptor. You don't know where those drugs are coming from or whether they're truly, USP pharmaceutical grade or not, all those kinds of issues.
And as far as I know, there's not really an easy way to get some of that compounding information. Can you request for the pharmacy to give you information about their drugs, like the compounding pharmacy, or is that not? I've never tried, so I don't actually know.
Where are you getting your drugs from, kind of thing? No idea if they'll give you that information or not.
Right, or like some kind of third-party inspection, because you hear about that with supplements, because that's the other big issue with the supplement market, is there's no real oversight. So what's on the label isn't what's always in the bottle. I would think compounding pharmacies have more oversight than that. But I've heard you can ask for third-party inspections with different supplements. Again, I haven't done it. So I was just curious if you knew anything on that end.
I think that varies by compounding pharmacy. So some of them will do more sort of quality control. And again, maybe it's through third parties, maybe it's internal. Obviously, they're not going to do it on every single pill that they make, or they wouldn't have any pills left, right? So they've got to do it in some kind of batch system, or intermittently or whatever. But I mean, I do think that's a legitimate question to ask when you're thinking about what compounding pharmacy to use. So what kind of quality assurance do you have in place, and who's doing it, and how frequently are you doing it, to know that the amount of stuff that you say is in the pill is actually in the pill? So great questions to be thinking about, when you're trying to figure out what compounding pharmacy you're going to use.
The other thing that we're seeing recently, or I don't know if recent, the last couple decades really, of different products coming out. There's the USDA approved and the FDA approved. And I think a lot of times veterinarians, we don't really know the difference. What would you like, just trying to tell people, like if a product says it's USDA approved, what does that mean as far as the process? Because like Tanovia had to go FDA. Is that correct? Like the approval process?
Correct. So USDA only has authority over biologics. So that would be vaccines and some of the antibodies. Theoretically, if there was some kind of cell-based therapy, that would potentially go through USDA as well. Drugs are the purview of the FDA, the Food and Drug Administration. So that's the way that gets sliced. So it's a little bit confusing when it comes to the cancer therapeutics market. So for example, so all cancer vaccines would go to USDA. Again, any cell therapeutics, if you had like some kind of activated cell therapy, or if you had a cell-based vaccine or whatever, that would go to USDA. But the place where it gets really weird and confusing is with monoclonal antibodies. So if you've got an antibody that you say works mostly by engaging the immune system, like an immune checkpoint inhibitor, for example, that's going to go to USDA. If you have an antibody that does not work that way, so let's say it blocks a receptor ligand interaction, like Librela, that's going to go to FDA. So it's, can you with a straight face say that this works by engaging the immune system? If the answer is yeah, you're probably going to USDA. If the answer is no, you're probably going to FDA. As far as the differences, the three big parts that you need for an application. So is it safe? Can you reliably manufacture it? And does it work? Are the same three bits, no matter which organization you go to. But, the individual rules are a little different. What you have to prove and how you have to prove it, and how long you've got to do it and how much it costs. I mean, those are all a little bit different. And there are sort of separate groups of consultants who have special expertise with one group or the other. And, you can find those people out there in the world who, might only work with USDA or might only work with FDA and stuff like that. But yeah, it's weird. It's weird and seems kind of arbitrary the way they sort of break some of those things down. But that's kind of the lay of the land.
In the USDA, that process for approval, is it as strict as the FDA? Is it as expensive?
It kind of depends on the year. So the USDA is kind of like, its pendulum has swung back and forth between being pretty lax to being like really, really stringent, and then, kind of going back and forth sort of depending on, I don't even know what, depending on the flavor of the month. So it kind of depends on who you ask and when, as to whether things are easier, harder, or about the same from one agency to the other.
Yeah, it is. That's what, when I've looked into some of this stuff just with consulting, it's really interesting, and just kind of the breakdown of who does what. And the other thing I think we've seen recently is some of the different diagnostics that have come out in veterinary medicine. They don't actually have to apply for approval. Is that correct? Or because in human medicine for a diagnostic, does it have to go through like FDA or USDA?
So the diagnostics on the veterinary side are completely unregulated. So it's like the Wild West. So buyer beware. On the human side, I think the big thing is if it's going to be covered by insurance or not. So if it's going to be covered by insurance, usually they want to see a pretty high level of evidence that it improves the outcomes, or they're not going to cover it. So often that's kind of it's not really a regulatory bar, but that's where the level of evidence needs to be, is if you want third-party reimbursement. There is this thing called CLIA, right? Which is sort of a laboratory certification, but that has more to do with, are the test results reliable and repeatable? Not so much what does it mean and does it improve the outcomes of patients? So that is one sort of bar that you can, attain for a diagnostic. But yeah, much more that is really governed by the insurance companies on the human side.
So on veterinary, it's really just up to the veterinarians, I guess. Like you said, buyer beware as far as some of that, where are products successful or not.
Yeah, pretty much anybody can hang out a shingle and start selling a diagnostic test out of their basement on the veterinary side.
That is interesting. I hadn't really thought of it out of the basement, but you're right. I guess you could start anywhere. So that is true. And, you're involved in a lot of the, I would say, high level stuff in vet oncology as far as the journal and, president of VCS. So what's some exciting things that you see coming up, and maybe also on the flip side, some concerning things that you're seeing?
The thing that I'm looking forward to the most is I think we're going to see a lot more stuff in sort of the biotherapy / monoclonal antibody space coming up in the next few years. So I think we're all familiar with the fact that there have been peer-reviewed publications about, an anti-CD20 antibody that Elanco has. Very, very recently, at the most recent VCS meeting, Zoetis talked a little bit about a CD20 antibody that they have. Obviously, there's this Merck monoclonal checkpoint inhibitor that's out there. There's other checkpoint inhibitors that are being developed. Again, there's this nerve growth factor antibody that Zoetis has, Librela, right? So I think we're probably entering an era that human oncology was entering like 20 years ago with monoclonal antibodies, where it's all of a sudden getting much more easy, thanks to the ability of smart people to like move genes around in cells and stuff like that, to actually develop these antibodies and, eventually sort of to produce them at scale. So I'm really hopeful that the next 5-10 years, we may end up seeing a lot more antibodies become available. And I think just, what we would refer to as like the naked antibody, just like a monoclonal antibody sitting there in a vial, is probably just the tip of the iceberg. And, the next step beyond that, and this is something that's getting to be really cool on the human side, are what are called bispecific antibodies, where, if we all remember back to our basic immunology, right? Most of our antibodies are like little Ys, right? So there's like, this little part here, and then there's this thing down here, right? And these parts up here are the parts that actually recognize stuff. And then the part down here is the part that like interacts with other cells and does stuff like that. Oh, hey, I got some balloons. And what the bispecifics do is actually there's one little part that is, recognizing your cell of interest. And maybe the other part is stimulating the immune system. So maybe it's binding to T cells or doing things like that. And again, on the human side, this concept of what are referred to as bispecific T cell engagers are really, really, really cool. So this is like, one way that you can actually target, a cancer cell and activate T cells at the same time. And maybe this is a sort of a sneaky way that we can do something that's very, very similar to, the cell-based therapies that they're doing in humans like CAR T, but with something that's off the shelf, in a vial that, we can give to our patients very quickly and easily. So that would be really exciting if people can start moving genes around to start looking at that sort of approach. The one thing that's a little concerning about that is there may be more toxicity that's seen with those guys than there are with just sort of like the naked antibodies, and whether that's something that, again, we as, the veterinary oncology community, and the companies are going to be comfortable sort of marketing again to that sort of non-specialist is going to be an interesting nut to crack. The only way we're going to know, like, well, how toxic is it? And, what kind of things do we see and how hard is it to manage is to do it though, right? We're not going to know that until we try. But I'm pretty fired up about that. I'm also super fired up about how cheap it's getting to, sequence DNA these days, right? And I think the amount that we're learning about that, sort of what do things look like in dog tumors and how close do they resemble the human tumors and things like that, is obviously super exciting, but it's also a little bit of a double-edged sword in my mind, right? So, yep, we all know how to sequence DNA now, and I certainly believe everybody who's out there who's doing it, but I think the thing that's problematic and where we need to really take that information with a grain of salt is extrapolating too much from, what's known on human side, which isn't necessarily even the same from tumor type to tumor type. So just because my tumor has mutation X, does that mean I should treat with drug Y? Not necessarily. And it might depend. The answer might be yes for certain cancers and no for other cancers. And we don't know any of that stuff. So just because we know how to find stuff in DNA doesn't mean that we know what to do with it. And that's, I think, going to be a much, much, much harder problem to solve than, again, being able to find the mutations. And that's probably something that's going to take a super long time and a lot of effort to get figured out. And the only way that we're going to be able to do is one drug and one mutation at a time. And that's a long, complicated, and expensive process to get through. But I don't see another way to do it.
Yeah, do you know if anyone's running clinical trials on things like that? Or are you guys doing any trials on anything like with genetic sequencing or anything?
So we're doing a lot of sequencing, but at least currently not, with an eye toward personalized medicine, because we're not there yet. So we've actually just finished or in the process of finishing a study looking at doing single dose pharmacokinetics, plasma pharmacokinetics of some of these target agents that people are starting to use. So, I mean, a lot of the drugs that people are using these days, we don't even know if we can get enough into a patient to do any good, right? So we're trying to crack that nut a little bit. Well, if people are going to be using these drugs, let's at least take a stab at trying to figure out if we're getting enough into a patient to be helpful. So, we're doing that, and, eventually we'll publish that data so people at least know, if I reach for this drug, do I have any hope of getting enough into a dog to do any good or not? So, but we're nowhere close to the point of being able to say, oh, well, if a dog's got this mutation, then you got to give that drug. I mean, we just aren't even close to being able to do that yet. We got to figure out the drug side of it first. In parallel, yep, we're sequencing a lot of dog tumors here. Dawn Duval's been doing that for years and years and years, and she's sort of continuing to sort of pick away at that. But I think putting those two things together is where it gets really tricky.
So it's hard to find, like, is the mutation the driver and will this drug work with it? That kind of thing. Is that what you're saying is the tricky part?
I mean, you can find a mutation in a tumor that's a driver for one cancer and it's not in the other. And, how are we ever going to figure that out? I mean, the best example of that on the human side I can think of is BRAF, right? So BRAF is a unequivocal driver mutation in human malignant melanoma. And drugs like vemurafenib work awesome. But you can also find BRAF mutations in human thyroid and colon, and vemurafenib doesn't do anything. Same mutation, same drug, completely different outcomes. And how are we going to figure that out? Like in our lifetimes? I don't have any idea. I mean, it's going to be really tough.
Yeah, it certainly sounds like an uphill climb with this. And, like you mentioned, trying to figure out, the dosing, like, can you get enough of a dose in to make a difference? That made me think of the losartan study where they found that we have to give way higher doses than what you do for your typical blood pressure, when they went back and looked at that data.
Right. I mean, you bring up a really, really excellent example there, right? So if, Dr. Dow said, hey, look, it looks like losartan can inhibit monocyte migration. And he just started giving the regular old dog dose to a dog without looking at the PK or the pharmacodynamics. We would have ended up giving 10 times too little losartan, and it would have been a negative study. It wouldn't have done anything, and that would have been the end of the story. So again, without that informative PK, you just never have any clue if what you're doing is any good. So yeah, thanks for bringing up that example. That's a great one.
Yeah, well, and it's one that I think about because I don't know about you, but often when I prescribe losartan, the pharmacists freak out. They have a tendency to tell owners like, this is lethal, don't do this. And it's like, no, I promise it's okay.
Yeah. We've had the same thing happen.
Yeah, but I mean, I guess it's good the pharmacist is paying attention, right? So I suppose, that's the positive for when we get our meds.
Yeah, totally.
And, another kind of thinking of the future, different things out there, what do you think about precision medicine and data-driven approaches, so like AI tools? How do you see that fitting in? AI is such a big topic right now, and just in general, where do you see it in oncology or maybe just vet med?
Wow. I mean, that's a really interesting question. And actually, a colleague of mine is actually trying to determine if there's enough interest in a standalone journal looking at artificial intelligence in veterinary medicine. So could that be like, is there enough stuff going on that med is ready for that? Not just oncology, right? But, across the field in general. I think that's really speaks to the fact that this is something that's going to be used more and more. I mean, in our sort of day-to-day practice, we're already starting to use it to abstract medical records. So like when we get RDVM records in, we have AI tools available that are like officially sanctioned, by the veterinary teaching hospital that we can actually use to abstract those records, to save people having to go through like 120 pages of medical records. So that we're starting to use, at least exploratorily, some of these AI-based dictation tools for doing case summaries and stuff like that. So I mean, that's here already. That's like in the clinic as we speak, which is pretty exciting. I think some of the other low-hanging fruit that we're probably going to see soon, obviously, there's companies that are starting to market some sort of AI-based tools for looking at blood smears and doing cytology and things like that. So I think that's going to be coming up pretty fast as well. I know there's some services that are starting to do the same thing with radiographs, right? So actually getting radiograph reads from AI. I got to be honest, I've only sort of encountered that a couple of times, and so far I'm not very impressed, but that's, going to be getting better as we move along. I think overall we're the things that worry me about sort of using it more broadly are, again, just the fact that it's this black box, right? So like nobody knows what these machines are thinking or how they're reaching the decisions that they're reaching. And I think in the medical community, that's scary, right? One of the things that I love when we're looking at a cytology or when we're reading a radiograph is you can go down and talk to the radiologist or the pathologist and be like, why'd you say that? Why do you think this is lymphoma and not reactive? And, at least here, right? In an academic center, you can just literally walk down the clin path and like, there's the clinical pathologist. And you can be like, can you show me that slide and talk me through it? And they're like, sure, no problem. And like, we are never going to be able to do that with AI, right? Like, we just have to trust them. And I think that's pretty scary for a lot of people still, like today, just that sort of letting go. Like, there's not a professional you can reach out to who can explain the why behind the decision making. And I mean, I think that's going to continue to be an issue on the diagnostic side, on the sort of more therapeutics or personalized medicine side. Like, to what degree are you going to trust a machine to make the final decision for your patient? And I think maybe that's going to be partly a generational thing, right? As sort of people come up having AI sort of more embedded in their daily life, they're going to be like, of course an AI is reading my radiograph. Well, duh, who else is going to do it? But, for us older people, it's like, wait a second, man, I'm not sure how comfortable I am with that. And, it's another time will tell thing. I think we'll end up seeing how comfortable clinicians end up being and how much control they're willing to sort of give up to these algorithms. So super, super interesting question. And yeah, really curious to see what the next 5-10 years are going to bring in that realm.
Because I think it'll be that fast too. I think in 10, 15 years, we could see big change. Because it was funny you mentioned the generation, because that just made me think back to, I've worked with clinics that fought against fax machines and then they really fought against email. So does that mean, am I going to be the one fighting with AI? I don't know.
Right.
Maybe.
It's totally true. I mean, it's coming really, really fast. And I hate to say it, and it doesn't just go for medicine, but I think it's going to be here before we're ready.
Yeah.
And
Right.
and we're not really going to know what to do with it for a while. I think, yeah, the technology is going to be here, I think, before the wisdom, maybe. We'll see.
Yes.
Yeah, I'm a doomsayer, right? I'm not saying we're going to let it take over everything and it's going to destroy humanity or anything like that. But yeah, I mean, that is a real worry, is maybe we're going to just let it do more than we should. And, we are going to end up making some bad decisions as a result, but we'll see.
Right. Yes. Well, and I think hopefully enough people making comments and saying like, hey, oversight and looking into things. Hopefully, like you said, it won't turn into the Terminator movies and
Yeah.
we'll be okay. But, this has been great. I always love chatting with you. It's so fun to catch up. I know it's been more than a hot minute since you were my mentor at Colorado, but it's certainly so great to see you. And, as we're wrapping up, I always like to ask people, who do you think would be a good person to have on this podcast?
Oh, I think a really, really interesting person who could really sort of speak to the clinical side and, what it's like running a big, extremely busy and complicated sort of academic oncology service would be our own fearless leader, Dr. Sue Lana. I think getting her insights would be really, really cool.
Yeah, no, that is great because that is a different way to look at it because you guys certainly run a huge service. CSU is very well known, and you guys have done a great job of keeping that, right? It wasn't, well, this was the main university for a few years. Like, you guys have done a really good job of setting the bar high and keeping it there, which is, I'm sure, not an easy task. So yeah, she would be great to talk to for sure. Well, again, Dr. Thamm, thank you so much. Really appreciate having you on the show today.
Thanks, Dr. Venable. Great to talk to you.
Well, that's it for this episode of the Veterinary Cancer Pioneers Podcast. If you enjoyed this episode and gained valuable insight, we would be so grateful if you could share our podcast with your friends and colleagues. And it would be even more wonderful if you want to give us a 5-star rating, positive review, or any kind of feedback on Apple Podcasts or wherever you listen. The Veterinary Cancer Pioneers Podcast is presented to you by Impromed.